Hello friends, in today's class we are going to study about the citric acid cycle. So the citric acid cycle is the aerobic cell metabolic wheel to generate energy by acetyl coenzyme A oxidation. Friends, as you can see in the screen that glucose get converted into glucose 6-phosphate that can undergo pentose phosphate pathway or can undergo synthesis of glycogen, or it can form pyruvate, which is simple glycolysis. Also the pyruvate get converted into glucose 6-phosphate through gluconeogenesis. We will be talking in details about the gluconeogenesis in our next video. Also, we can see that pyruvate undergo to form lactate and ethanol, or it can form acetyl coenzyme A. This acetyl coenzyme A is a connecting link between this Kreb cycle and the glycolysis. This citric acid cycle is the final common pathway for oxidation of fuel molecules like amino acid, fatty acid and carbohydrates. Friends, it is the fuel molecule enter the cycle as acetyl coenzyme A. So this cycle in the red box is basically the Kreb cycle. This cycle was discovered by Hans Adolf Kreb in 1937 and he got Nobel Prize in 1953 for physiology. The site of occurrence of this cycle is inside mitochondria. As you can see on the screen that from glycolysis, the end product of glycolysis, which is pyruvate, that converted into acetyl coenzyme A with the help of pyruvate dehydrogenase. And to do so, it follows some steps, in which step 1 is the pyruvate, a carboxylic group is removed from pyruvate, and this is the carboxylic that this gets removed and form carbon dioxide. During this process, one molecule of NAD positive get reduced to NADH, and then a 2-carbon compound is formed, which is attached to coenzyme A, called as acetyl coenzyme A. This acetyl coenzyme A enters into the Kreb cycle. The overall reaction is pyruvate plus coenzyme A plus NAD gives acetyl coenzyme A plus carbon dioxide, because the 3-carbon compound is getting converted into 2-carbon compound, and a NADH plus H positive released. You can see the reaction involved in the conversion and the enzyme responsible for it. We are not going in detail to study this step only. So let's begin with the citric acid cycle. The citric acid cycle is also called as Kreb cycle or tricarboxylic cycle and is the hub of metabolic system. It accounts for the majority of the carbohydrate, fatty acid and amino acid oxidation. It also accounts for majority of ATP generation of these compounds and others as well. It is a amphibolic pathway, which means it acts as both catabolically and anabolically. The overall reaction undergoes like 3 NAD plus FAD plus GDP plus phosphate plus acetyl coenzyme A changes into 3 NADH plus FADH plus GTP plus coenzyme A plus 2 carbon dioxide molecules. So let's see the overview of citric acid cycle. In this cycle, here is the acetyl coenzyme A. A 4 carbon oxaloacetic acetate oxaloacetate condenses with 2 carbon acetyl to form a 6 carbon citrate. An isomer of citrate is oxidatively decarboxylated and 5 carbon alpha ketoglutarate is formed. Then alpha ketoglutarate is oxidatively decarboxylized to yield a 4 carbon succinate. Oxaloacetate is then regenerated from succinate. 2 carbon atoms acetyl coenzyme A enter the cycle and 2 carbon atoms leave the cycle in the form of molecules of carbon dioxide. 3 hydroxide ion, 6 electrons are transferred to 3 molecules of NAD+, 1 pair of hydrogen atom, 2 electrons is transferred to 1 molecule of FAD. So let's see the cycle. As we can see here, that pyruvate get converted into acetyl coenzyme A, which reacting with oxaloacetate to form citrate. Citrate undergoing series of reaction to form isocis aconitase, then isocitrate, alpha ketoglutarate, succinyl coenzyme A, succinate, fumarate, malate, oxaloacetate. During the course of this path, it is removing one water molecule, then it is accepting one. The NAD is getting converted into NADH and carbon dioxide molecule is released. One more carbon dioxide molecule is releasing and NADH is getting produced. One ATP is produced. One FADH is produced, FADH2. One more water molecule is consumed, and here one more NADH is produced. So let's see the structure of this cycle. Here we can see that acetyl coenzyme A is reacting with oxaloacetate to form citrate. Citrate is releasing the water molecule because it having the negative charge over here, and this negative charge will force to form the double bond with the alpha carbon. This will be the formation of isomer. Then the cis aconitase will undergo to form isocitrate, where again the water molecule will react and it will get attached to alpha carbon. Here then it will again form a isomer and will make alpha ketoglutarate. During this process, one NAD will get reduced to NADH and one carbon dioxide molecule will be eliminated. remove. Then again this double bond ketone will be unstable, will react with coenzyme A to form succinyl coenzyme A, and again one CO2 will be obtained and NAD will be reduced to NADH. Further, succinyl coenzyme A will react with phosphate and water to convert one GDP into a GTP. Again, when this succinate will be converted into fumarate, it will give us one FADH, FADH2. Again, fumarate will react with a water molecule to give malate. After this, the citrate, we can see here citrate with the help of aconitase get converted into isocitrate. Isocitrate with help of isocitrate dehydrogenase get converted into alpha ketoglutarate. These are enzymes that will be responsible for the change in configuration or the conversion of compounds. Then alpha ketoglutarate will be converted into succinyl coenzyme A with the help of alpha ketoglutarate dehydrogenase complex. Succinyl coenzyme A will be converted into succinate with the help of succinyl coenzyme A synthetase. Succinate will be converted into fumarate with the help of succinate dehydrogenase, and fumarate will be converted into malate with the help of fumarate. Malate will be converted into oxaloacetate with the help of malate dehydrogenase. And then finally the oxaloacetate will be converted into citrate again with the help of citrate synthase. Let's talk about all enzymes one by one. If we look at the properties of citrate synthase, citrate formed by acetyl coenzyme A and oxaloacetate with the help of citrate synthase. Only cycle reaction with C-C bond formation, because we have seen that 2 unit of acetyl group attached to 4 carbon unit of oxaloacetate unit to produce 6 carbon compound, which is called as citrate. Here you can see the reaction. Oxaloacetate is reacting with acetyl coenzyme A to form citrate. Now let's see about aconitase. Its role is elimination of water from citrate to form carbon-carbon double bond that we have seen of cis aconitase. It is stereospecific addition of water molecule to cis aconitase to form isocitrate. Here is the reaction. Citrate get converted into cis aconitase and then again to isocitrates. Now come to isocitrate dehydrogenase. It is oxidative decarboxylation of isocitrate to alpha ketoglutarate is a metabolically irreversible reaction. One of 4 oxidation reduction reaction of the cycle. Hydride ion form the C2 of isocitrate and transfer the NAD plus to form NADH. Oxalosuccinate is decarboxylated to a alpha ketoglutarate. Here is the reaction. Isocitrate gets converted to oxalosuccinate and then alpha ketoglutarate. The alpha ketoglutarate dehydrogenase complex. It is in this enzyme similar to pyruvate dehydrogenase complex. Same coenzymes, identical mechanism. It also has three enzymes: alpha ketoglutarate dehydrogenase with TPP, dihydrolipopyl succinyl transferase with flexible lipoamide phosphate group, and enzyme 3 is dihydrolipoyl dehydrogenase with FAD. In the reaction, you can see alpha ketoglutarate plus NAD plus coenzyme A in presence of alpha ketoglutarate dehydrogenase get converted into succinyl coenzyme A, and one carbon dioxide molecule is obtained. Also, NAD get reduced to NADH. The succinyl coenzyme A synthase. The free energy in thioester bond of succinyl coenzyme A is conserved as GTP or ATP in animal or bacteria or plants also. Substrate level of phosphorylation reaction. Also, the succinyl coenzyme A on reaction with succinyl coenzyme A synthesis form succinate. The succinate dehydrogenase complex. It is the complex of several polypeptides and FAD prosthetic group of iron sulfur cluster embedded in the inner mitochondrial membrane. Electrons are transferred from succinate to FAD to form ubiquinone in electron transport chain. Dehydrogenation of stereospecifically from the trans isomer is formed. Succinate get converted by succinate dehydrogenase into fumarate. If we talk about fumarase, it is stereospecific trans addition of water to double bond of fumarate to form L-malate. Only L isomer of malate is formed. In this, fumarate get converted into malate using fumarase enzyme. The last enzyme is malate dehydrogenase. Malate is oxidized to form oxaloacetate with the help of this malate dehydrogenase. So if we look at the stoichiometric tree of the citric acid cycle, 2 carbon atom enter the cycle to form acetyl coenzyme A. Then 2 carbon atoms leave the cycle in form of carbon dioxide. 4 pair of hydrogen atoms leave the cycle and 4 oxidation reaction. 3 molecule of NAD and 1 molecule of FAD are reduced. 1 molecule of GTP is formed. 2 molecule of water are consumed. So let's see what we have gained in the reaction. So if you will ignore this glucose to glucose 6 phosphate and fructose to fructose 1,6 biphosphate, just ignore all this. We will start from here. The pyruvate gets converted into acetyl coenzyme A. It will give 2 NADH and isocitrate converting into alpha ketoglutarate will give 2 NADH and similarly alpha ketoglutarate will give 2 NADH. Then further these energy are obtained. Here it is 5 because we are considering it into 2.5 ATP of energy. If when we will study about the electron transport chain, there are the complexes through which they form to pump the hydrogen to form NADH. Here I have mentioned how much ATP will be formed of one NADH, FADH and GTP. Okay, so the functions of citric acid cycle, if we talk about, we have already seen that it is amphibolic, so it will undergo catabolism and anabolism. Anabolism, it will be starting for many reactions, and catabolism will come from carbohydrate, lipid and amino acid. Yields energy in form of GTP, yields reducing power in the form of NADH and FADH. This regulation is done by allosteric modulators, covalent modification of cyclic enzymes, supply of acetyl coenzyme A. These will regulate. Also, the enzymes also have the inhibition that will regulate these cycles, like citrate synthase is allosterically inhibited by NADH, ATP, succinyl coenzyme A, citric as a feedback inhibition, whereas isocitric dehydrogenase, the allosteric inhibition are NADH, whereas ADP will increase the promote the citric acid cycle. Okay, then alpha ketoglutarate dehydrogenate complex will inhibit by ATP, succinyl coenzyme A, NADH. Here you can see on which step whatever is producing, basically if that thing exceeds in number, the cycle will get stopped. So the positive sign is promoting, whereas negative sign is inhibiting the steps. Now finally, Krebs cycle is a source of biosynthetic precursors. We can see many of the things can be formed from different products of Krebs cycle, like citrate can form fatty acid and sterols, alpha ketoglutarate can form glutamate and other amino acids, and ultimately purines also. Succinyl coenzyme A can form porphyrins, heme part and chlorophyll, whereas oxaloacetate can form aspartate and which will form purine, pyrimidines, amino acid and other complexes, whereas oxaloacetate can also undergo gluconeogenesis. Phosphoenol pyruvate and ultimately will form glucose. We will see in the next video about this. Thank you.